Hello, so today I'm going to be discussing the errors that occur during the replication of DNA. So, DNA replication errors is a very natural and common occurrence in the process of DNA replication. So, as we know, DNA is made up of the double helix structure and is made up of four basic fundamental bases, and these bases are connected to a phosphate group and a deoxyribose sugar molecule. So, these bases are adenine, thymine, cytosine, or guanine, and each one of these bases has a pair that it gets paired to when talking about the double strand helix. The adenine base pairs with the thymine base, and then the guanine and cytosine bases pair. So these are the usual pairings of the bases. But these usual pairings can get broken and messed up, which is called a tautomeric shift, which is the first DNA replication error that I'm going to be talking about. And this is the idea in which normal DNA base pairing shifts, and now the bases are paired to the wrong base. So I researched an article discussing tautomeric shifts, and the title was DNA Replication and the Causes of Mutation. And the article talks about how purine and pyrimidine bases of DNA exist in two different tautomers. So basically they either happen when the nucleotide bases are shifted from their normal base pairing to the rare tautomeric pairing, or these rare base pairing arrangements result when one nucleotide in a base pair is in the rare form. So essentially, in easier terms for me to understand even, is let's say guanine bonds, its usual bind is to thymine, or its usual bind is to cytosine, but instead it bonds to thymine. That results in a replication error and has consequences. So a different form of replication error, moving on from tautomeric errors, is the idea of insertions or deletions in nucleotide bases. So in some rare cases, the newly synthesized DNA strands can result in the addition of a nucleotide base that shouldn't be there. This is the idea of insertion. And on the other hand, there's a different type of mutation called deletion, where full nucleotide bases can be deleted and taken out. These three concepts, insertion, deletion, and tautomeric shifts, are three examples of errors in replication in just the structure of DNA, and these specific examples all have consequences. So a specific consequence for the insertion mutation could be the loss of function in certain proteins, and it's a frame shift mutation because you're adding that nucleotide that shouldn't be there. And then on the other hand, if a deletion were to occur, genetic diseases can happen because the deletion could take full genes and delete them from the nucleotide base or amino acids that are needed for a proper genetic function. Some of the consequences of the tautomeric shifts that I talked about, it can be a lot of different small things, but some of the most interesting ones that I researched were where I found an article titled Structural Insights into Tautomeric Dynamics in Nucleic Acids and in Antiviral Nucleoside Analogs. So this article basically talks about the idea of the tautomeric shifts in DNA and its consequences. And as I said earlier, tautomeric shift in DNA is when there's a mutation allowing certain nucleotide bases to change function and structure. These shifts that happen due to the tautomeric shift can have a lot of consequences, and some of the main ones that I found in this article were that it can cause spontaneous mutations during DNA replication, and it can also reduce the success rate at which DNA replication occurs, and like the overall genetic mutations. And there's a lot of overall genetic mutation that can occur from the tautomeric shifts, and these have a lot of effects on the DNA and the cells as a whole. So those are the specific consequences of those three specific errors in replication that I researched. But another example that I actually learned last year in biology class was that errors in DNA replication can even cause cancer. And there's a specific gene called a tumor suppressor gene, and if this gene is too active in the cell and in the DNA, it can cause cells to overgrow and lead to uncontrollable cell growth, which is basically where the cell just divides at such a rapid rate that it causes cancerous cells. And that's a very severe consequence of a DNA replication error that affects a bigger proportion of the body rather than just the DNA or cell. The last thing I'm going to touch on is the question about whether enzymes in the cell work to fix all these errors. So from what I've found, basically no, Enzymes cannot fix all of these errors, but they do fix some. And one of the examples is tautomeric shifts. There's a specific enzyme. How the tautomeric shifts is basically the process of nucleotide mismatching of the nucleotide bases. And the tautomeric shifts have a specific enzyme called the mismatch repair enzyme that can come in and essentially repair the bases so the bases match up again. And that's one enzyme in specific that can help the tautomeric shifts. So bringing it all together, essentially I only talked about a few of the errors that can happen in replication, although I know there are many more errors because DNA replication is such an intricate process and errors are bound to occur naturally. But those are the few that I researched, and I hope you enjoyed.